All right, welcome back, everybody. In this lecture, we're going to be discussing a very important structure that allows our minds to function. In particular, we're going to be looking at a cell that is the main catalyst behind all of these functions called a neuron. We'll explore in this lecture what neurons are, how they're structured, and what it is that allows them to communicate with each other in the very elaborate ways that they do in order for all of our mental processes to unfold. But to understand the amazingness of the neuron, I first want to take a journey back to a time before we had a lot of the scientific tools that we have at our disposal in order to study the mind in the neurological ways that we do. If we go back a couple hundred, in some cases even 1,000 years, people were still interested in the mind, but they didn't have a lot of the devices that we have at our disposal that give us the ability to talk about things at a cellular level and even understand all the structures and how they're built the way we can do it now. So what people did back then, hundreds or thousands of years ago, is just postulate models and ideas that could help people tackle some of the things related to the mind that they were interested in. For example, there were people historically that were interested, just like they are today, in individual differences and emotions and where these things were coming from. And it eventually led to a very enticing idea that's still in circulation today called the humors theory. It was this notion that our personality and our emotions were tied very strongly to the balance of different fluids that we had within our system. You had the typical blood that you would think of when talking about fluids in the system. You had phlegm, which we don't like to think of, but is also a fluid. And then you had these other two things that were a part of the humors theory called black bile and yellow bile. The notion was that if you had too much of something in your system, either overall or just in a moment, you would theoretically see a shift in your demeanor, in how you reacted to situations, and a preponderance of something might even shift a person as a whole. There were probably some reasons for why this particular idea was in circulation for a long time. A great example of this humors theory that maybe helped it continue progressing, even if it wasn't necessarily a very sound idea, was this notion that if you had too much blood in your system, you were something called sanguine, and it led you to be extremely talkative and energetic. And to balance things out, all you needed to do was let a lot of blood out of the individual, and things would predictably calm them down. You don't have to be a neuroscientist to know that, yes, if you do drain the blood of a lot of individuals and there isn't blood flowing through our system providing oxygen to lots of things, lots of parts of our body are going to start shutting down. This is obviously just a one-off example of where the humors theory might have worked, but I want us to recognize when talking about it that it was something that was kind of needed in attempts to understand the body and the mind when we didn't have a lot of other mechanisms at our disposal. This is also true for people trying to explore our motor system. Numerous theories and ideas popped up over the different centuries that we wanted to understand how we were able to feel and experience all the sensations that we were experiencing and then react to those things in kind. One of my favorite examples of this is something called the hydraulic theories for system, or the hydraulic theories idea that proposed that our touch and our movement systems were both run through some type of hydraulic chamber, much like an engine for a train or other mechanism, where changing air pressure, changing fluids, and kind of their preponderance in different areas might be what allows us to feel things like heat or to send messages from the brain to the body. Honestly, this theory is pretty darn intuitive, and there are reflections of this notion of the brain having pathways from the body to the brain and from the brain to the body that we still recognize today, just not necessarily with this belief that there's channels of air or fluid that are going back and forth between these two things that allow touch and movement to actually occur. Another really popular area of interest that people tried to get at from a number of different perspectives was this mind-body problem that was talked about in the first chapter of your book. How we could separate out the mind from the body, or whether or not these two things were integrated, was not only a source of debate, but people were trying, when we didn't have a lot of tools, to figure out how we could verify different ideas. There were individuals actually measuring people on their deathbed, trying to see if they could detect when they died the removal of the soul by seeing just a fraction of their weight drop at the moment they died. There were individuals trying to find ways to tease apart the soul from the brain by doing biopsies and other things within individuals. Some of these approaches were definitely gruesome, but it really highlighted how, even if we didn't have a lot of tools at our disposal, people were still really interested in the mind, and they wanted to find a way to figure out what made it tick. Well, fortunately for us in science, in the mid-1800s, we started to develop a lot of tools that were allowing people to look at all the different biological mechanisms of not only the human body, but many different species. And what we were able to do when we started breaking down the body was recognize that we had all of these different systems, like our circulatory system, our muscular system, our skeletal system, that were operating not only on the structural level, but the cellular level. And it led to a flurry of theories and ideas about maybe there being some type of a system that also allowed our brain to communicate with the body, even if we couldn't see it as easily as something like our circulatory or our skeletal system. And it eventually led to a theory that maybe a large portion of our mental world was actually just dictated by fairly basic communication of cells that were given the name neurons by a famous theorist named Heinrich Wilhelm Waldier. This idea of neurons, once it started to get proposed, started to catch on, and people started to try to figure out how a simple cell could actually allow us to do all the elaborate things that the mind did. And there was a theorist named Ramon Cajal who looked at all of our other systems that we had throughout our body and proposed that, much like all those other systems, our nervous system, which is what he called it, could potentially allow us to actually do all these elaborate things through just very simple communication. So he set out to describe what he called the neuron doctrine, that laid out how this nervous system, which he called it, might actually work. He argued that we'd need millions or billions of these cells throughout the body, most of them concentrated in the brain, and that they would need to communicate with each other through small gaps or some type of mechanism that allowed them to kind of move freely while some way forming junctions between cells. And after Cajal and Waldier presented these ideas, many people in biology, many people in psychology, which was sort of a budding science at the time, started trying to find ways to discover these neurons. One particular biologist by the name of Camillo Golgi started tinkering with different alloys, different types of stains that he would expose small slivers of flesh to see if he could detect these undiscovered cells that we were already calling neurons before we knew they actually existed. And eventually, Golgi came up with this combination of silver and other alloys that we now call the Golgi stain, that gave us our first images of these really cool, intricate cells that we, again, to this day, call neurons. You can see in the background of this slide an example of what a neuron looked like through these Golgi stains. They essentially looked like big giant spider webs with sort of a hub connected to them that seemed to reach out into different areas and have all of these connections with different cells within this thing that we were now calling the nervous system. And after the work of Golgi and the ideas of Cajal and other people that flocked into neuroscience, we started to really try to dissect what this nervous system entailed. And today, we now, about 150 years later, have a very good understanding of what our nervous system is all like. One of the first things to note is that Cajal was really correct in the fact that most of our mental world does seem to be dictated by these small microscopic cells that we call neurons, and we do indeed have a lot of them. Most estimates suggest that by the time you're a college student, you probably have roughly 80 to 100 billion of these cells located in your body, with the vast majority of them being found in your brain, and in particular, the structure in the back of your brain called the cerebellum. This kind of high concentration of neurons is not the only place, though, where neurons are located. You also have a number of them reaching out to your periphery through systems called the peripheral nervous system and other structures that are kind of connected to it. Some of our neurons are very tiny, even for cellular sizes. Others actually stretch vast spaces to get messages from things that are in our very far peripheries, like our toes and our fingers and other areas, to things like our spinal cord and other structures that are part of what we call our central nervous system, something we'll be talking about a little bit more in our next lecture. And what we find when looking closer at these neurons is that they do communicate in very basic ways, but because there's so many of them and because they're connected in such elaborate means, they are thought to be the things that allow us to do all these different functions of the mind that we talked about in that first slide and that we continue to explore in psychology to this day. So what are these structures? What allows this neuron to function? There's a lot of critical things that we want you to be familiar with so you can better understand multiple components to our nervous system. The first place I want to start when talking about these neurons is the inside of them, the hub that's a part of pretty much every cell that we have in our system, regardless of whether or not we're looking at our nervous system, our circulatory system, our respiratory system. Essentially, every cell that we have in our body usually has this thing called a cell body. And that cell body contains a structure called the nucleus, which is critical in the ability for these cells to survive and to react to the world they're designed to react to the world in. Now, connected to those cell bodies are little tiny branches that we call dendrites. Now, some neurons have multiple dendrites, others have just one, but these dendrites typically reach out from the cell body and connect to either adjacent neurons or connect to specific areas within the body. Usually we're talking about sensory cells when they're not connecting to other neurons, so they can pick up on information, collect information that's then collected by the nucleus within the cell body, so the cell can decide how it wants to react to that information. And when we're talking about reactions, how a cell is going to kind of, interact with the environment, most of the reactions that we tend to look at, even though there technically are some other ones, relates to the other major structures in this cell, in this neuron. The first major structure that we probably want to focus on is the long strand that extends from the cell body called the axon. Now, you can have multiple dendrites in each cell, but each cell in our body has just one axon. And the structure that connects the axon to the cell body is often called, as you can see in this picture, the axon hillock. It's a really critical juncture to take note of when we start talking about how these cells actually propagate messages from the cell body out to the ends. That's actually the next place I want to mention. So as we progress from the cell body through the axon out to the ends, we get to these structures that are sometimes called synaptic terminals. Other times they're called terminal buttons. But essentially what you're looking at here are these buds on the ends of these cells that form off of these things called terminal branches. So you've got one axon that can kind of reach out to a specific area, and then at the end of the axon, usually a number of different branches that reach out to other cells or other structures within the body so they can send some type of a message. And to understand how these messages are sent, we have to look a little bit closer at those terminal buttons. So within those terminal buttons are these really interesting collections of chemicals called neurotransmitters. They're essentially the messengers for cells when they want to talk to adjacent cells or body structures. And the way they communicate with each other is through these different chemical reactions that happen in a microscopic space called a synapse. Sometimes you'll also see it listed as the synaptic gap or the synaptic cleft. And when these cells communicate with each other through the use of neurotransmitters, what they're essentially doing is sending some type of chemical compound that reacts to the dendrites or adjacent cells or structures in some way that do one of two things. They either excite the adjacent cell, getting it to be much more likely to act, or they inhibit the adjacent cell, getting it much less likely to act. Now, usually when people think of neuron communication, they think of the excitatory message. Now, we want to see these cascades, these lightning effects happening when cells are connecting with each other. And to be honest, a large portion of the cellular activity that we see in our body is excitatory in nature. But we don't want to overlook the inhibitory messages that are being sent out all the time as well. Oftentimes, these inhibitory messages are kind of counterbalances to the excitatory messages that allow the excitatory messages to be more efficient. A classic example of this is if you say, lift your arm up. When you do that, you've got a whole bunch of excitatory messages going from your brain, down your spinal cord, out your arm, to that shoulder that allows you to raise the arm by flexing different muscles on the top of your shoulder, and potentially even on the top of, say, your bicep or tricep, depending upon which part of your arm you're lifting. But while you've got all those excitatory messages going on, you also have some cells that are trying to start eventually an inhibitory message that goes out to other muscles in that arm. If you reach up on your shoulder while you're raising it, you'll feel flexed muscles, but if you slide your hand down to the under part of your arm, no matter how much you exercise, I guarantee you'll feel a lot of muscles that are completely deadened. It's kind of a counterweight. It's actually impossible to flex both your top and bottom muscles at the same time because of the way these inhibitory messages work. And mind you, there's a lot of excitatory messages traveling down your brain, down your spinal cord, down to the area beneath your arm. But then when you get to the cells communicating with the muscles, they at that point don't send an excitatory message anymore. They send an inhibitory message, which gets those muscles to essentially turn off, not be something that can be activated. And that's just one example of these inhibitory messages. Another one I love to talk about is the inhibitory messages that are always going on in your brain, essentially shutting portions of the brain off so it doesn't get overwhelmed. I know many of you might have seen those stupid movies where people take chemicals or get injected with something and magically their whole brain is on at any point in time. They talk about how they're using 100% of their brains. That would be extremely dangerous. You would probably die from that because you can't have every area of your brain active at any given point in time. It just doesn't function when that occurs. You need to have big portions of it turned off so you can process what it is in front of you. Having that inhibitory message allows you to do that. There's also something that's really critical in things like sleep and other components to our mind where we really don't want everything functioning at any given moment. This inhibitory message, again, though, is just half of the messages that can be done through the neurotransmitters that are released in these synaptic clefts or synaptic terminals or synapses. And it's really important to note, again, that the excitatory message is a little bit more common than the inhibitory one. But understanding these messages is a really critical part to understanding how neurons do what they do. It's just not the only thing we want to understand. And it's also not the only thing we need to understand if we want to understand how people today have the ability to study the brain in very complex ways. Another thing that we have to take a glimpse into to understand how the brain works and how we're able to do all the amazing things with the mind we're able to do is this process that we call the action potential. Essentially, it's the conveyance of that message from the cell body to those terminal buttons that we talked about before. A little bit about the action potential, if you've never heard about it before. It's an all or none message that is sent from the cell body that starts at the axon hillock, works its way down the axon, gets out to the terminal branches, and eventually results in the release of those neurotransmitters into synapses that we talked about in the previous slide. Now, this action potential does not vary in strength within the cells. As the kind of statement implies, a cell is either going to fire or it's not. It doesn't really fire or not really fire. When we're talking about magnitude of effects, it's not about the strength of the cellular reaction. It's about the quantity of the cellular reaction. So if something's really loud or really soft, you don't experience something as being really loud because you've got cells screaming at each other. You simply have a lot of cells communicating when it's loud and not many cells communicating when something's soft. So this message, this action potential, as I said, is an all or none response that gets propagated along the axon and eventually allows neurotransmitters to be released. But what happens for this message to propagate itself down the axon? Well, that was a mystery for a long period of time. And it wasn't until we were able to not just get pictures of these cells, but look even deeper through much more advanced microscopic devices, that we were able to see that this kind of process, exchange, actually happened on the ion level. So what we see when we look very closely at the axon hillock and the structures along the axon are these tiny little pustules that are located throughout these axons. And these pustules are just large enough to allow the exchange of sodium and potassium ions to come in and out of them at different points in time. So when a cell decides to fire, it's because the dendrites have received enough excitatory or inhibitory information. Well, in this case, it's fire. It's received enough excitatory information that the cell body decides to send a message. The way it sends a message is by having a tiny pustule, a collection of tiny pustules, I guess, located at the axon hillock to open up. And that allows a bunch of adjacent cells in the environment that are positively charged to come rushing into the cell body. And these positively charged ions that come rushing in due to an imbalance of pH inside and outside the ions cause kind of an overload of the cell. And in order to kind of balance things out, to get things less cluttered, there's another pustule, another channel right next to the one that opened up originally that opens up and allows a number of other ions, potassium ions in this case, to go rushing out of the cell. And that second door that opens actually triggers the opening of a third door, which allows new ions, new positively charged sodium ions, to come rushing in. And as you might guess, there's then another reactionary door that allows a bunch of other ions to go rushing out. And this opening and closing of doors, this pattern, it actually propagates itself all the way down the axon until it gets to those terminal buttons when, for the very first time, you no longer have the exchange of ions that were continuing to occur, but instead you have that release of neurotransmitters. It might seem like a very tedious task, but this actually happens in a fraction of a second. Usually we're talking about measuring things in milliseconds as to how long it takes for a message to get from that cell body out to those terminal buttons. It's a very effective way of having these messages stay consistent from the cell body to the terminal branches, no matter how long these cells are. They can be millimeters long or even shorter, or they can be inches or even feet long in terms of the length of the axons, and still get the message across just as robustly, even if it maybe takes a little bit longer to travel those really long distances. The knowing about this is just kind of cool on its own, but you'll understand a little bit later when we look at how we study the brain in the next lecture, that there's a lot of utility in knowing about this information as well. Another thing that we want to be aware of is that when there is communication between cells in that synapse, there's a lot more than just this process of what we call activation that occurs. Activation is essentially the excitation or inhibition of the adjacent neuron due to what the neurotransmitters are doing. In addition to that activation, another really cool process that often occurs once a cell releases neurotransmitters is something called reuptake, where the cell that had the terminal buttons filled with neurotransmitters that were released actually finds a way to suck up a large portion of the neurotransmitters that weren't broken down or collected by the receiving cell. Now, some of us might think of reuptake as kind of a random, weird thing, but if you look at it in efficiency level, you can appreciate why reuptake is so unbelievably important. First big plus to reuptake is it allows cells to not have to constantly be producing more and more neurotransmitters. It's essentially recycling at its best. But another big plus to reuptake is it allows those synapses to be cleared of clutter, so the communications between these cells can happen at a very rapid pace. We're talking about cells firing and then firing again in fractions of a second. To aid this reuptake process for that purpose, another thing happens within these synapses that we sometimes call diffusion, or you might see it described as metabolization, where there's different enzymes and chemicals that can actually work their way through those tiny microscopic synapses that we talked about earlier and kind of remove all the clutter and, in particular, all the neurotransmitters so they don't accidentally inhibit or excite a neuron after they were supposed to. This rapid diffusion and metabolization of the cells is usually a good thing, but it can cause problems when maybe cells aren't allowed to communicate as effectively as they should. In fact, lots of drugs, which we'll talk about later, have been designed to kind of slow down this diffusion metabolization process, and in doing so also slow down the reuptake process. It doesn't necessarily stop it, it just slows it down to an extent, for a fraction of a second, so cells can maybe communicate a little bit more effectively at the cost of maybe sometimes leaving that message out there a little too long. I know we've covered a lot, but there's one last thing I want to discuss that actually is not really highlighted very much in your textbook. It kind of bothers me to an extent because it gives us this impression that our nervous system is really just a bunch of neurons running around doing magical things without any assistance whatsoever. If you look closely at our nervous system, what you find is that our neurons are actually greatly outnumbered by a collection of other cells within that system that we called glia, or sometimes you'll see listed in other places as glial cells. Now, these cells are designed to really help and support the nervous system and the neurons that are doing all the amazing things that they're doing. And the way they support this system is multifaceted. One set of glial cells that we have are called astrocytes. You see them pictured in the lower left-hand corner out of the three pictures. These astrocytes allow these neurons to stay structurally sound, to kind of have nice clean paths between them, and also they're designed to remove debris if, say, a neuron dies or something gets in the way of the communication between neurons. They're like tiny little cleanup crews that help really make sure that neurons are doing what they need to do. Another collection of neurons called oligodendrocytes and Schwann cells, two very different looking types of glial cells, can help these neurons do something really cool called synchronization. Now, we've talked so far about how communication happens at a singular cell level. So if a cell wants to communicate with another one, it sends that message down the axon to the terminal branches, and then that synapse is released to just one other neuron. That's a very simplistic description of how cells actually communicate. Remember, we've got billions and billions of neurons within our body. So how do they work the way they do? Well, we usually have multiple neurons connecting to multiple cells, branching off to a number of different areas. So you've got these really intricate webs that are designed. So when cells communicate with each other, when the cells are going to be activated, very rarely is it just a one-to-one correspondence where one cell is going to communicate with another, who's then going to communicate with another. It's usually much more intricate, where multiple cells are coming together to try to communicate with multiple cells, who then come together and try to communicate with multiple cells. So how do we get these messages to be more effective? And this is where the glial cells come into play. Many of our oligodendrocytes and Schwann cells, well, actually many of our oligodendrocytes, not Schwann cells for this matter, will wrap around multiple neurons, wrap around their axons, and in doing so, they can coordinate the nervous system responses of them. They can get the action potentials to occur at the same exact moment. So the messages can be really strengthened for the receiving cells. If we were in class right now, we do this weird activity that everybody doesn't quite understand, but I think does eventually get my point across, where I have the entire class first just clap at their own pace. Now, whenever they want to clap, just clap while we're sitting in class. And what you get is kind of a low murmur of clapping that isn't necessarily going to overwhelm anybody. Then I ask the class to clap just as loudly as they did the first time, but to do it in a coordinated way, where we all clap together. And what you find, much like you find at a sporting event or other place where people do these unison claps, is that immediately when we all clap at the same time, the message gets much stronger, much easier to pick up on. That's the benefit of these oligodendrocytes. It allows whatever the message is, be it excitatory or inhibitory, to be much more likely picked up on by the adjacent cells. But the other thing oligodendrocytes do, which relates to the other types of glial cells that I already briefly mentioned, called Schwann cells, when wrapping around these cells, they provide something called myelination. Myelination, or the myelin sheaths, are actually the byproduct of the fatty structures of oligodendrocytes, or the entire bodies of these Schwann cells, wrapping around different segments of the axon of neurons. In many of our what we call myelinated cells, multiple Schwann cells have wrapped around different segments of the axon. And you might ask yourself when looking at that, why? We can sort of understand that that probably plays a role in how oligodendrocytes get these cells to synchronize with each other, but why would it be beneficial to have just this wrapping in general? Well, it does theoretically provide a little bit more protection for these cells from debris and other things. But the main reason why this myelination process takes place is because by doing it, it actually speeds up the cellular communication process. As we talked about earlier, for a cell to communicate with another, it commits this thing called an action potential. The way the action potential occurs is through the opening and closing of those tiny pustules that are happening or propagating all the way down the axon. When a cell's myelinated, what it does is allows those cells to not have to open and close doors in the structures where the fatty substance, where the myelination, is wrapped around those axons. A great example of how this works is one where I actually line up students in my intro classes and have them play a game where they're kicking each other in the feet or tapping each other on the shoulder, depending upon how I'm feeling that day, in some type of long line. So I'll start at the back of the line, tap somebody in the shoulder, they'll tap the next person on the shoulder, and so on until you get to the very end. It's essentially what's happening when an action potential occurs. Right? You've got one activity that triggers another activity all the way to the end. And what we do in this activity is we measure how long it takes for us to get from me tapping the first student on the shoulder to the very last student getting their shoulder tapped. And then the second part, to kind of mimic what this myelination does, we separate out the groups. So we take the entire line of people and have it split into two lines. But we ensure when doing this that the length of the lines, not the number of people in them, but the length of the lines are identical to what they were when everybody was just part of one line. And then we have these two lines again. Commit this activity where somebody's tapped on the shoulder or kicked on the foot, whatever we choose to do, and it goes from one end to the other. And we measure how quickly this happens. And what we find every time is that it's not necessarily cut in half in terms of how long it takes to get from one end to the other, but it is reduced substantially as to how long it takes to get from one end to the other, even though we're traveling the same distance both times. And that's exactly what happens here with this myelination process. You get some awkwardness as those cells are trying to get the messages down the axon with these kind of myelinated sheaths wrapping around them. But by having these myelinated sheaths wrapped around, the message can work its way down a little bit faster down that axon. And that myelination is something that is really critical for a lot of different processes of the mind. If you don't appreciate how valuable it is, just watch a 2 or 3-year-old try to do something like play baseball, where they're trying to track a ball, and by the time they're able to send the messages from their brain to their arm to move it, after messages have gone from their eyes to their brain to kind of coordinate how things are moving in the first place, usually by the time their arm started to move, the ball's either hit them or already gone past them. A big reason for that is this lack of myelination. To be really coordinated, to be able to function at an efficient speed, you need cells to communicate even faster than they originally can. We might think of fractions of a second. Milliseconds is more than sufficient, and it is for many different mental processes, but there are some that require even more efficiency, and this is where these glial cells really come into play. They allow these cells within our body to be as efficient as possible. It's a little awkward, but it really does help. One last thing I want us to focus on before we close out, and I promise we'll be revisiting a lot of this stuff later on, are those chemicals that these neurons are releasing into the synapses, those things we called neurotransmitters. When we first discovered them, we thought of them as fairly interesting. That we essentially have different chemical compounds that seem to be getting some cells to be active while getting other cells to be inhibited. As we've continued to study neurotransmitters, though, what we've tapped into is this really amazing world that's much more diverse than we had originally anticipated. In the beginning, we thought we'd just find 2 types of neurotransmitters, the excitatory and the inhibitory ones. But now we know that we have around a dozen different neurotransmitters within our system, and we've got other chemicals that can impact cellular activity as well that are often linked to this nervous system. And these neurotransmitters usually are kind of concentrated more in specific cells than others, even though we now know that certain cells contain actually multiple types of neurotransmitters. And they tend to react with certain cells in specific ways, even though there's certain cells that do react to multiple neurotransmitters in different ways. And that kind of diversity of neurotransmitter types and kind of ways they're clustered have led to some really interesting discoveries that have kind of focused on where concentrations of neurotransmitters seem to be found in different parts of our nervous system. We don't have the capacity in a class like this to cover all the different neurotransmitters that are there, but I do want to focus our attention on just 2 of the ones that are really important to know about, simply because they have so much to tell us about what we've discovered with respect to neurotransmitters. The first neurotransmitter I want to talk about is one that most of us should be kind of familiar with and want to be even more familiar with. It's a neurotransmitter called dopamine, or you'll see it listed in many scientific journals as DA. Usually when we talk about dopamine in psychology, we tend to be discussing its implications in reward and kind of positive motivational experience. And that's because you do have structures within the center of your brain, part of something called the limbic system, that do secrete massive amounts of dopamine when we're experiencing pleasurable things. Remember, when we talk about secreting dopamine, we're talking about lots of neurons that use dopamine as their primary means of communication being extremely active when we're experiencing something very positive or motivational. And what we found is that this high level of dopamine activity in these areas is not only something that we usually immediately recognize as pleasurable, but it's something that activates other systems within our body, like ones linked to memory and ones linked to attention. It's also important to note, though, with dopamine, that this is not the only thing that this neurotransmitter has been tied to. We also see dopamine being used by a large number of other structures of our nervous system that go beyond just this limbic system. There's also research that's shown that dopamine levels are linked to a lot of our different sensory systems within our brain, what we call the projection areas. We see that high excessive amounts of dopamine activity are often tied to hallucinations, schizophrenia, and other things where our sensory systems aren't functioning correctly. We also know that having too little dopamine can lead to a lack of connections between critical motor systems that are tied to diseases like Parkinson's disease and other types of diseases where our coordination is no longer functioning at the right level. Essentially, what I want you to see with dopamine, just by sampling a small portion of the things that this neurotransmitter has been tied to, is that this is a very multifaceted neurotransmitter. You'll hear people talking a lot about dopamine and how important it is for us to feel good and to love things, but know that too much dopamine can be a problem for other systems, and too little can be a problem for not just our mood and our happiness and our attention, but it can impact even our physical world as well. This is also sort of the same for most of our neurotransmitters, but each of them has kind of their own unique story that we like to tell. One I always like to mention in intro class, because I know many of you might never take a psychology class again, is a neurotransmitter we'll revisit again when we get to the clinical side of things, called serotonin. It's often listed in textbooks as 5-HT. Serotonin is traditionally tied to psychological wellness and mood. There's many drugs that have been devised over the years to increase the activity of serotonin-using neurons to try to help boost or elate moods for people struggling with anxiety or depression issues. It's also important to note that serotonin can be triggered by more than just specific drugs. There's numerous studies that have shown that varying levels of activity, changing your diet, changing your sleep cycles, can all impact the functionality of neurons using serotonin. In essence, what we see is if you start messing with large portions of your life, you start to mess with the internal structures of your body, down to the cellular level, to where they're more or less active. And in doing so, it can have an after effect on things like your mental functioning. Numerous studies have suggested that the precursor to symptoms of depression tend to be things like dramatic shifts in diet, the different changes in your sleep cycles, stressors that can get certain areas of your brain to start turning off. And when they turn off, it starts to become very difficult for people to come back from those problems. But shifting those things can help ameliorate some of those issues. The kind of cyclical nature of this is something that many people have looked at and really examined once we started to understand the importance of this particular neurotransmitter and all the systems that are tied to this. Again, I just want to reiterate, these are not the only two neurotransmitters that we have in our body. They're just the two that we're going to highlight in this lecture. If you want to learn more about them, I encourage you to start looking in the next section, looking into the readings that we have, and potentially even considering taking some type of neuroscience class and a drugs in the brain class, so you can really appreciate how much amazing things we've discovered when it comes to neurotransmitters and the other chemicals that are used by the neurons within our body. Well, this is where we're going to end, but I do want us to just kind of get some context around what we're covering. I wasn't trying to just rush us through neurons and then move on to the next thing. I really want to always start this intro class by covering neurons, because if we understand them, then we can better understand the brain and the rest of our nervous system. And if we can understand that, well, then we can better understand lots of different components to the mind. The reason why we dwelled on lots of very specific things going on is because what I want to cover in our next class, in addition to looking at the structures of our nervous system, is how we know what we know about those structures. And a lot of the mechanisms and things that we focused on here will pop up again when we talk about those things in our next lecture. But for now, this is a good place to close out. So hopefully you got a lot from this. And I'm hoping that I'll see many of you soon when we get into our very next lecture. Make sure that you're reading, make sure that you're going over those systems, and, well, hopefully I'll see you soon. Bye now.